For this video, I'm going to show how to analyze a gene set with other gadgets in Garuda once the data has been retrieved from Opentox. And this assumes that you've already gone through the steps in my first video, so you've discovered samples and you have retrieved data into the data fetcher. So for this one here, I'm looking at still acetaminophen in vivo rats, liver, high dose, 24 hours. And I've set the p-value filter now to 0.001. Though it's not so important exactly what it is, it's just that we want to have a manageable size of gene set, not too big. Then I'm going to click on the gene column, and only on the gene column. That's going to be the only blue column. And then I'm going to make sure that data is selected in the top right corner. And then I click discover. And this will show you all the gadgets that can accept a gene set, which is quite a few, and they can do various kinds of things with these gene sets. So first off, we're going to sound this gene set to the Reactome gadget. So I double click here. And that launches the Reactome gadget. So Reactome is a pathway database. So it's going to take our genes and test all the pathways for enrichment. So we can see now there were several matches. So these are the most significant pathways in that gene set. Unfortunately, these are Homo sapiens pathways and Homo sapiens genes. So the conversion was done based on the gene symbol only, which is quite rough. So we may hope that in the future, hopefully, there will be a better version of this gadget that will distinguish between different species, and that's also something we have to do on our end. But you have these pathways here. They can be exported, and if you click on one of them, such as glucuronidation, then it will open a web browser page with the more extensive information about this pathway and the Reactome website. So that's Reactome, so that's one way to characterize your gene set. So next I'm going to go back to the data fetcher. So here is my gene set column. I click discover again. I haven't changed anything. And now I'm going to click on the Persilome. So double click there. So Persilome is another transcriptomics database developed in Japan, but slightly different properties from Opentox-Garuda. So what I do here is first I select a gene of interest, say ACBD6, and then most of the data in Persilome is actually for mouse, so I'm going to stick with mouse. I select a chemical and a project I care about, and I click get data. And then here I see actually a time and dose response curve for that gene in mouse for this compound. And I can also click here and inspect in detail the numerical data. So we can try with a different gene, let's say GLRX2. And here is the time dose response curve for that one. Now, because the server is located in Japan, occasionally this database is not accessible from Europe or not easily accessible, but I hear that the team is working on improving access. But this is an interesting potential comparison study to compare with the Opentox-Garuda's response. Next, I go back again to the data fetcher. I've selected my gene symbols. I do the discovery once more. Now I'm going to select Panther Analytics. So Panther Analytics is able to discover many concepts associated with our genes, such as geoterms and pathways and proteins, and so on and so on. So to use this, first we have to select a gene. Let's say we select CREG1, click on search, and we wait a little while. And now we have discovered and characterized proteins and some geoterms and some families. And if we double click on an object here, it's going to open again an official web browser page from PantherDB, which is going to show more information about that entity. So that is also another interesting source of additional information. So there are many gadgets that can receive a gene set and explore it in various ways. So if you're interested, please play with Garuda yourself and explore it and see what you find.